Right. Right. So what is DNA technology? DNA technology encompasses both biotechnology and genetic engineering. It involves the study and manipulation of DNA molecules. Through this technology, scientists are able to analyze, modify, and replicate DNA sequences. With that information, various practical applications can be enabled. And that technology can be used in many fields like agriculture, medicine, forensics, environmental science. And we'll be touching on a few in a bit. So this is what DNA looks like in the case that you've never seen what a DNA strand looks like. I'm sure everyone sees that. Because once it comes to biochemistry or biology, you always see a graphic um, of DNA. But this is what it looks like on a molecular level, showing you the different base pairing that occurs. So the first thing I wanted to touch into was going towards the DNA replication, just understanding how our DNA replicates. DNA replication is a process by which a cell makes identical copies of its DNA before cell division. It's a crucial step in cell reproduction, ensuring that each cell receives a complete set of genetic information. So if you weren't aware, all your cells, well, except for your neurons, have to go through the cell cycle. But before it does that, you have to replicate your DNA. So our DNA strands are held together by hydrogen bonds, the bases. So we'd have an enzyme called helicase that would first break the hydrogen bonds between the strands and basically open it up. So now you have two template strands that would then be replicated. And feel free to stop me anytime to ask any questions, by the way. But now we'll go into the steps. So like I said, first we have the unwinding of the double helix, and that is done by the enzyme helicase. It breaks the hydrogen bonds between the base pairs. Then there is template strand. So each separated strand now serves as a template for building a new complementary strand. We would then have complementary base pairing. So now that we have the template strands exposed, free floating nucleotides in the cell's nucleus are attracted to the exposed bases. And then we build a new strand. So then the enzyme called DNA polymerase moves along the template strands and it adds complementary nucleotides one by one. So then we form two replicated DNA molecules. Once the DNA polymerase is completed its work, two new DNA molecules are formed, each consisting of the original, which is the parental, and one newly synthesized strand. So 5 simple steps involved in the replication of DNA, and that is an important theory because it helps you understand how we use PCR. Now, PCR is a technology I'm sure many persons were exposed to during the pandemic because when you went for COVID tests, well, initially it was mainly PCR being done, but then we started doing rapid tests. But so today we'll go into just how PCR works and the application of PCR. PCR can be used not only with the diagnosis of diseases, it's also used in many other fields related to biology and also forensics as well. But PCR is a powerful tool used to amplify samples of DNA. Due to its specificity, it can be applied to several fields, like I was saying. So to run a PCR experiment, you would need primers. So primers are genetic material, like usually single-stranded DNA, but they're what identify, like that's what you use to identify your sample. So when we go into the like how it works, you'll see the importance of primers. Polymerase, as we saw in DNA replication, polymerase is the enzyme that will be adding the nucleotides when we go in the strand. And obviously we need a sample DNA. So that's when you use, so I'm just using the PCR test, for example. So when they push the swab in your nostril, is to get some DNA sample. So that is what they would use to run the PCR test. Then you have a thermal cycle, which is used to run the PCR experiment multiple times and change the temperature. Because for PCR to occur, there is a lot of changes in temperature. So we'll go into it so you see like how the temperature changes. DNTPs, these are free nucleotides that will be used to grow the strand. Obviously, for an enzyme to work, we have multiple enzymes, but for enzyme to work in this experiment, you need a buffer system to give it the optimal environment. And also another thing that enzymes need is cofactors. In this case, we would use magnesium ions. And also we could use some additives and enhancers to make the experiment more efficient and effective. So let's go into the steps of PCR. The first step is denaturation. So you would first need to break the hydrogen bonds between the DNA in our sample. Now, in DNA replication in your body, the enzyme responsible for that would be helicase. But in a PCR experiment, what we would do is we would break the hydrogen bonds by increasing the temperature. So when a PCR experiment starts, you would raise the temperature to 95 degrees Celsius. At that temperature, the hydrogen bonds that hold the strand together would break. So now we're denaturing, so we're breaking that hydrogen bonds. Now we have two strands. If you look on this side, so this was the original DNA strand with the hydrogen bonds. You have a question? Oh, hi, good morning. So yeah, we have the DNA sample here, and when we raise the temperature, what happens is the bonds will break between the base pairs, and now we get two strands, right? So now the next step would be annealing. We would reduce the temperature at this point to about 55 degrees Celsius, and what this does is it allows the primers to bind onto, well, it's complementary base pairs. The primers would now bind onto so if you look here, the primers will now bind onto the strands that we just created. Now that is granted that that the primers recognize the DNA strand. So for example, when you do a PCR test for COVID, if you have the virus present, then the primers would recognize that genetic material, and it would bind in this step. If you don't have COVID and you do a COVID test through PCR, then the primers would not anneal because the primers are complementary. So we know in biochemistry, the way base pairing works is guanine to cytosine. So that's C2G and A to T. So these are the complementary base pairs. If the primers don't anneal, then that means that there was no viral material in that individual. So that's why when you do a PCR test, there are times when during the pandemic, I remember people were saying that you had the flu. They thought that they had the flu, and when they did a PCR test, it would come back positive, and they were like, well, once you have the flu, you have COVID. But that's not how it works, because the influenza and SARS-CoV-2 has two different genomes. So if you do a PCR test using primers for SARS-CoV-2 or COVID-19, then it's impossible that you had the flu. But that is the annealing step, where the primers would bind onto the strands, the template strands that we just created. And then the next step is the extension step. And that is where our polymerase would come in, and it would extend this. So starting from the primer, it would extend it. So it would build a strand. So it would continue to put nucleotides in here. Now the nucleotides it would add in are these DNTPs that I said were necessary. These were the nucleotides that are in solution, in our solution. And they would add them one by one and grow the two strands. So that is how it would work. One thing you should know is that for the extension step to happen, we have to for the extension step to happen, we have to raise the temperature back up to 72 degrees Celsius. That is a temperature where DNA polymerase, well, in this case, the type of DNA polymerase that's usually used is Taq polymerase. So for, I think you could see it here in this diagram, it's just Taq polymerase. Yeah, this is a polymerase that we would use, and it works at an optimal temperature of 72 degrees Celsius. So these are enzymes. If you do any basic biochemistry, you would know that for enzymes to work, they need to be in the right pH, they need to be in the right temperature for them to work optimally. So that is where we would bring the temperature back up to 72 degrees Celsius. And that really concludes the three major steps in a PCR experiment. These steps are repeated multiple times. So that's why you would use a thermal cycler, because it needs to continuously run those steps. So the denaturation, annealing, and extension steps are repeated multiple times in a thermal cycler, a device that automates the temperature changes required for each step. So in PCR, these are the three temperatures that you have to keep in mind. So 95 degrees Celsius, why? To break the hydrogen bonds between the base pairs for us to get two template strands. Then we have to reduce the temperature to around 55 degrees Celsius, and that would allow our primers to anneal. So basically bind complementary to the template strands that we just created. For this step again, if the primers don't recognize the DNA sample, then that means that it's not complementary. So you basically have a negative result. But if it binds, if the primers recognize that DNA sample, then that means that it is a positive step. Now, like I said with PCR, it's done to amplify a DNA sample. So we started off with one template strand. However, when you've done a PCR, you realize that you now have two strands. So because we got one, we broke it up, we got two templates, but now we grew those templates. So now we have two identical strands to the first one. And since this is continuously happening in a cycle, what would happen is this would keep amplifying. So now that we have two, the same thing would happen again. So then we'll get 4, and then we get 8, and we get 16, and it continues. So you run that cycle 20 to 40 times. And what you realize is that you like amplifying it. So you're making more of the same sample, granted that the primers recognize it. And that's how you would get a positive result. So that's the theory behind PCR. Any questions on PCR? Okay, so I don't see any questions. So, and PCR is very specific. So you could have the smallest sample, that's very effective in forensics, where someone leaves us very small, like from their hair, like just a strand of their hair. And we could use that genetic material to run a PCR result experiment. So it's very specific. Once the primers are able to bind onto that template, that sample DNA, then it could be amplified, because we'll keep making more copies until we have enough genetic material to run some analysis on it. So applications of PCR, disease diagnosis. So it was done a lot during COVID. There's also forensic analysis, genetic testing, genetic engineering. So yeah, that was a blank slide. Yeah, to remove that slide. But now we're going to look at for PCR and forensics, amplifying DNA to investigate analysis. So PCR is a powerful tool used in forensic science to amplify tiny amounts of DNA found at crime scenes. It has revolutionized the field of forensics and its applications in criminal investigations. So like I was saying, a lot of the time, criminals could leave little genetic material at the scene. But because of the way PCR works, we could actually use that to make more copies of that sample and be able to analyze it better. And there are different ways that you could do this. You could have DNA profiling, because PCR enables the creation of DNA profiles unique to the individuals. These profiles obtained from biological samples, so like hair, blood. So you could have a drop of blood, you could have a strand of hair, a fingerprint. Serves as a crucial evidence for identifying suspects, victims, and establishing familial relationships. Challenging samples, PCR sensitivity allows analysis of degraded DNA from older evidence or difficult samples such as bone fragments or teeth. Crime scene samples, forensic investigators collect biological samples like blood, saliva, hair, or skin cells from crime scenes. PCR allows amplification of trace amounts of DNA, turning minute samples into sufficient quantities for analysis, like I've been saying. And STR analysis, short tandem repeats analysis, is commonly used with PCR-based techniques in forensics. STR loci are regions of DNA containing repeated units. PCR amplifies these regions, and the lengths of the repeats are used for profiling. So that is something that is used all the time. That's something that's always used. Short tandem repeats right now. With forensic genealogy, if you're interested, you could look into that. One application that type of forensics was used in was to find a Golden State killer. He was a serial killer in Golden State, and they never caught him until he was like 80 something years old, and it was because through the use of STR analysis, they were able to find him by linking his relatives' genome with his. They were able to narrow down potential suspects, and they had a sample of his DNA from back in the day. I think he did his crimes in the 80s, 90s, but they were able to match his DNA with a sample they obtained when they narrowed him down as a suspect, and then they got a tissue that he left behind. It's a very interesting case. You could look at the report on YouTube. Just shows how forensics is really growing with the advancements in technology, particularly PCR and genetics. But now I want to move into gel electrophoresis. If you've worked in a lab, if you've done any lab work, a lot of time we do gel electrophoresis. I'm not sure if any of you have experience with that, but gel electrophoresis is a fundamental technique used to separate and analyze DNA fragments based on their size and their charge. It's widely employed in research, forensics, and genetic analysis. So for the setup, DNA samples are loaded onto a gel matrix, often made of agarose or polyacryl. I'm going to butcher those names. That's the one thing in biochemistry I don't like. It's just the long names. But then you run an electrical current, and if you're aware, your genetic material does have a charge. So when you run it for an electric field, so you run an electric field across the gel, and that causes the negatively charged DNA fragments to migrate towards the positive charge. So because of the phosphate that is found in our DNA, it does have a charge. So when you run when you run an electric field on the gel, it causes the fragments to move, and smaller fragments will move faster than the larger fragments, and then so like they separate based on their size. And because of each and every person has a different genetic makeup, you would get different results. So then you could use that as a comparative method for identifying individuals, for example, or whatever test that you're running, you could use that. So this is what it looks like. So we would load the DNA samples in here, and then we apply the charge. And what happens is it will move down based on the size. And as you could tell, like we have different, so these are 3 different individuals, and then you get like um, different results. Yeah. So based on that, you forming a profile for someone. So if you run this and you form one profile, and then you get another sample, let's say the next 12 years, and then you run it again and you get the exact results, then that means that there is some similarity between the genomes of these two individuals. And obviously you could, for confirmatory purposes, you could go in and do further testing, but it's a great way to identify, like just to compare. So applications of this technology would be DNA fingerprinting. Unique binding patterns on a gel can identify individuals for forensic and paternity testing. PCR product analysis. So PCR amplified DNA fragments are visualized to verify successful amplification, sorry. Genetic mutation detection, mutated DNA segments exhibit different migration patterns. This could also be done with not only DNA, but you could also run it with proteins. I remember one of the experiments I had to do at university was testing for proteins. So what we do is we use the amino acids, and the amino acids have, well, the charged amino acids have different charges. So then you would run it and then you'd be able to signify which one was what. So when something has a charge, you could use this technique. So now we'll go into DNA sequencing. And DNA sequencing is the process of determining the precise order of nucleotides in a DNA molecule. So it's a foundational tool in genomics, genetics, and molecular biology. And there are various types of sequencing techniques that exist. So there is Sanger sequencing and there's next-gen sequencing. Sanger sequencing was like the early technology that was used. Right now, next-gen sequencing is way more widely used because it's way faster, and it's for a machine, like you don't need to do anything manually. But I have a diagram just showing you what Sanger sequencing looks like. So we run a PCR with fluorescent chain ending DDNTPs. So what a DDNTP is, it's an NTP, it's a DNTP, sorry, but it does not, so, but when it's added to the chain, you cannot add on another nucleotide. So remember with DNA sequencing, well, not DNA sequencing, with DNA replication, what happened was the DNTPs will continuously added to form the chain. But if you use a DDNTP, what happens is when it's added to the strand, it stops there. Like it can't continue because it's lacking the necessary molecule to continue, for the chain to continue growing. So that's why it's called chain ending, chain terminating. So you have an original DNA sequence amplified for PCR, and then you make a mixture of DNTPs and the fluorescently labeled DDNTPs. So what happens is you grow a strand, but then when you, so this strand is growing, but when you added this DDNTP, it stopped. Then this strand was growing, when you added this one, it stopped. And that would continue. And then what happens is through gel electrophoresis, remember the theory we just did with gel electrophoresis, the larger ones would, the smaller ones would travel longer. So what this means is that the longer chain, so this one is the longer chain, where's my mouse? This one is the longer chain, so it would stop here. Then this one is the shorter chain, so it would add up. And what happens is that is the way that you would basically sequence the DNA sample that you had in. So it's based on the size. So just to reiterate, I'll try to make it as simple as possible. But we start with our DNA sample. And what happened is we ran it through PCR. So we have multiple copies of that sample, right? So now when we add in our DDNTPs, they're chain terminating. So once it's added, the chain cannot continue growing. It stops there, right? So when it stops, that will be our largest fragment. So when we run that in a gel electrophoresis, it would be to the top because it's the largest one. So it won't travel far. And then we have the other one, like it would continue that way. Then we excite it with a laser, and then we detect the first sequencing machine. And that is how it was done before, how you would get your sequence. But now with next gen sequencing, you don't necessarily need to do this. You just put it in the machine, I would run it and give you your genetic makeup. And that is important because for plants and even in us to identify the genes we have. Yeah. Also, if you want to do like, especially with plants, if you're doing genetic engineering and you want to see like if you change a nucleotide, what the outcome would come in the plant, that is also important. So it's a very effective tool. But now we're going to go into the interesting part of the science, which is really growing right now, and that is CRISPR-Cas9. I would have spoken about that yesterday briefly to the class yesterday when we were speaking about um, human genome editing. So when we spoke about the doctor who um, made changes to babies using CRISPR-Cas9 and he was arrested for that because there aren't really any regulations set in place as yet. But he edited a gene to make them immune to HIV, but he got arrested for it. But this is a technology that he used. So CRISPR-Cas9 is a groundbreaking gene editing technology that allows scientists to precisely modify DNA sequences within living organisms. Its potential for therapeutic application, genetic research, and agriculture has transformed the field of biotechnology. And CRISPR is very, like it's not complicated in the sense of what it's made up of. It has a CRISPR array, which is a clustered regulatory interspace short palindromic repeat, or CRISPR for short. And these are unique DNA sequences that act as genetic memory for past viral infections. And there's a Cas9 enzyme, which is an RNA-guided endonuclease that can cut DNA at specific target sequences. So that's why we realize that it's important for us to sequence the DNA. So now we know what the strand is like, and we could target that specific part of the genome if we know what the strand is made up of. So like I said, it was found in bacteria. This was a technique, a defense mechanism against bacteriophages. In case you've never seen a bacteriophage, this is what it looks like. It's kind of similar to, I know the sci-fi movies use these to identify like aliens, but it's really a natural virus. But this virus does not affect humans, it affects bacteria. That's why it's known as a bacteriophage. This is where its genome lies. So this would land onto the bacteria, and this is like a spring. So it would literally inject its genome through here into the bacteria. So a lot of sci-fi movies that were made revolving this virus. But CRISPR, which stands for, again, clustered regulatory interspaced short palindromic repeats, was initially discovered in the genomes of bacteria. It allows bacteria to remember and recognize previous encounters with specific viruses. So when a bacterium is exposed to a virus, it incorporates a small piece of the viral DNA into its own CRISPR array. This creates a genetic memory of the encounter. So yesterday for immunology, we did memory cells. So this is a mechanism used by bacteria that kind of mimics memory. So they incorporate part of the virus's genome into their CRISPR array, and that creates a memory of the encounter. So if the bacterium is attacked by that same virus, it uses CRISPR at the CRISPR array, which was the part of the bacteriophage's genome. So it uses that as a guide to recognize and target the invader's DNA. So it incorporates part of so it was infected by a virus. It cut part of the virus genome. It incorporated it into its CRISPR array. If it's ever attacked by that same virus with the same genome, what happens is it would quickly come in and cut that virus, and it wouldn't be able to replicate. And there are many applications of CRISPR, gene editing. So CRISPR enables the addition and deletion or correction of specific DNA sequences. Functional genomics, researchers can knock out or modify genes to study their functions. Disease modeling, CRISPR creates cellular and animal models to study genetic diseases. Therapeutic potential, CRISPR has applications in treating genetic disorders by editing disease-causing mutations. Agriculture, CRISPR can engineer crops for improved yields, disease resistance, and nutritional content. So the technology exists, like I was saying to the class yesterday, the technology exists. However, the issue is with regulation and ethics. You find that this technology is more used in plants because people are okay with us changing plants, killing off plants. But when it comes to animals, that's when you call in for animal rights, and also for humans, you say human rights, and there's a lot of ethics involved. So right now, advancements are mainly being made in plants more than in animals, humans. It's been used like we use like human cells to try it out, but to say on a large scale where we go enter clinical trials of making edits to the human, like a living human being, it's not yet to that point. Um, but it's very interesting that we could use that technology that was found in bacteria to um, work in our genome. So just to come back on this, what scientists would do is they would design the guide and RNA to target the part of the genome that you want to edit. And what would happen is once it recognizes once it's complementary, so once that guide and RNA is now complementary to the DNA, the enzyme would now cleave that area and allow the genome to now fix itself. So by doing that, so if we sequence the genome and we know exactly what part of the gene causes the mutation. So for example, we have a mutation at the CFTR gene, which causes some cystic fibrosis. So when we create a guideline RNA for that specific part of the gene, then we could cut that out and then replace it with a now healthy part of the genome. And that person will no longer develop cystic fibrosis, just for an example. But like I said, the technology exists, but it would take some time and regulations for it to be applicable on a large scale. And then there's also the part about who is it accessible for, the costing of this, richer individuals would would more be able to pay for something like that because the technology is expensive. So, but now we get into genetic engineering. Genetic engineering is a powerful tool in biotechnology that involves manipulating the DNA of organisms to modify their traits or create new ones. It enables us to alter the genetic information within an organism cells, offering a range of applications in various fields. So basic principles of genetic engineering: the isolation of DNA. So DNA containing the desired gene is extracted from a source organism. Then we have gene insertion. The target gene is inserted into the DNA of the recipient organism. And then we have expression. So the inserted gene is activated within the recipient organism, leading to the production of desired protein. And that was a bit of a mouthful, but there are different techniques that we use for genetic engineering. There's recombinant DNA technology, and I will show you a slide on what that looks like. So the DNA from different sources is combined to create a recombinant DNA molecule. We also have gene cloning, where recombinant DNA is inserted into a host organism, creating multiple copies of the desired gene. And then we could use DNA gene editing, where we use CRISPR-Cas9. So techniques like CRISPR-Cas9 allow for precise modification of specific DNA sequences. Now, I think, give me one moment, I'll pull up a video for us to watch on CRISPR, because I feel like that would give a better explanation of it, so that way you could visualize what I just said. Because I know just looking at words on a slide is difficult to picture how it works. So give me one moment. Let me pull up the video and then we'll continue from it. I have one more slide. Oh, yeah, I could just finish and then we'll watch the video after. But this is what a typical recombinant DNA looks like. So we have the human cell and we extract the gene from the human cell and we cut off that gene with sticky ends. So it depends. So we use endonuclease, which is an enzyme. So this time the enzyme we used was EcoR1. So we'd use it to cut off the ends, like flagging the gene that we want. Um, and then we would use a plasmid that is found in some bacteria, and we would use the same enzyme to cut this plasmid. So now we have sticky ends. So the sticky ends are like, so remember DNA is double-stranded. So if we cut it off right here and right here, what would have is would have some bases that lack a pair. Now that is energetically unfavorable. Like these bases need to be paired for it to be energetically favorable. So what happens is it's very easy for these two to come together. Because if you look here, this end here, and this end here. So what happens is they would come together and then you'd form your recombinant DNA. Now, a lot of these plasmids have parts of them that are able to express those genes. So what happens is when you insert that into a bacterial cell, it is able to express this gene. And that technology is used a lot. I'm not sure if you're aware of this, but we use bacteria to now create insulin. So before we would make insulin, we'd take insulin from pigs and other animals, but now we could use this technology to create insulin, and we could make a larger amount of insulin because it's bacteria, and we could have several bacteria. And all they do is you just continue expressing the gene that we took here, and they're using their expression machinery to create the protein that we're looking for. So that's what you call an expression vector. Um, there are a lot of them out there. I remember we did a lot of that. I remember at university looking at different expression vectors and different genes that we could put into the plasmid. But this is just a basic example of what it looks like. So I'm not sure if you have any questions. Feel free to stop me or interject. But yeah, it's pretty interesting what we could do, and obviously this opens up a lot of potential, because like if we could express more genes, then it could solve a lot of problems that we have. For sure, it solved the insulin problem, because now you don't have to rely on pigs to produce insulin. We could just use bacteria in a lab to keep pumping out the insulin for us. So I don't see any questions. So give me one moment to pull up the video on CRISPR-Cas9 just so you visualize how it looks. Okay, it's not a long video. So let me share my screen. Okay.